The Starling curve plots ventricular end diastolic volume, or preload, you can think of it as preload, on the x-axis, and stroke volume, or cardiac output, on the y-axis. The curve helps us visualize the interaction between preload and stroke volume. The way we're going to understand it is we're going to understand stroke volume as a function of end diastolic volume. You can see simply, if we are looking at the normal curve, as preload increases, stroke volume or cardiac output increases as well. This is the main concept of the Starling curve. It demonstrates to us graphically that stroke volume is directly related to ventricular end diastolic volume in a patient with a normal working heart. So why is this? Well, let's remember back to our myocyte cellular anatomy. The preload stretches the cardiac muscle fibers so that the actin and myosin filaments more optimally overlap to generate force. So if you think back to the way that the cell is oriented, here we have our myosin, and here we have our actin filaments kind of overlapping here. Thread, sort of a knitwork here. When there's more volume, or when the end diastolic volume is greater, we see that the sarcomeres actually spread out a little bit, so we get optimal interaction between the myosin and actin. So one way to think about it is that the more end diastolic volume there is, the greater interaction between myosin and actin in the sarcomere, which enables a higher contractile force and a greater stroke volume. So if you think about it just like a rubber band, the more you stretch the rubber band, the more it's going to bounce back. Similarly with the heart, the more you stretch the sarcomeres to a certain extent, the greater that it will recoil and contract and increase its stroke volume. So here we see the normal in a normal heart, but we also see that there's different situations, such as exercise and heart failure, where we see changes in this Starling curve. So we can see here specifically in heart failure, we see that this curve significantly changes in a way that when we increase our preload, we see only a slight increase in stroke volume, but certainly much less than normal. And then even at a certain point, we see as the preload increases, the stroke volume actually goes down. And we'll talk about that more in the pathology section. But the key thing to notice here that in situations of heart failure, when there's systolic dysfunction, when the heart is not contracting properly, we see that the stroke volume and the end diastolic volume relationship is disconnected. It's not normal. And that is this key downward shift that is seen in the Starling curve. So what's this heart failure and digoxin curve? Why is it up and behaving a little bit more closely to normal? Well, in order to understand this, we need to think about how digoxin helps our contractility. So let's take a moment and discuss how digoxin helps contractility of our cardiac cells. So the key thing here is that digoxin competes with the extracellular binding of potassium on the sodium-potassium ATPase on the cell of a cardiac cell. By doing so, it decreases the ability of the ATPase pump to push potassium into the cell and sodium out. It prevents this from happening. As a result, we see an increase in intracellular sodium. What does this have to do with calcium? Well, as we see an increase in sodium, we see a change in the sodium-calcium antiport. So we can see that as we increase our sodium, we'll increase our sodium going out here, which results in an increase in calcium coming in. So instead of sodium being exchanged for potassium in the sodium-potassium ATPase, we see sodium being exchanged for calcium in this sodium-potassium antiport right here. So that's how digoxin results in an increase in intracellular calcium. It blocks this sodium-potassium ATPase to drive calcium into the cell in exchange for built-up sodium. This increased calcium results in increased troponin C conformational change, resulting in more tropomyosin available to be bound by myosin. We see an increase in calcium, resulting in an increased binding of troponin C, which increases contractile force in our cardiomyocyte, which is known as inotropy. Inotropy is the concept of contractility. So when we say we have an increase in inotropy, we have an increase in cardiac contractility. We have a decrease in entropy, we say we have a decrease in cardiac contractility. So we can see here that by doing so, digoxin results in increase in our stroke volume curve, our Starling curve, so that when we have an increase in our stroke volume or increase in our end diastolic volume, we have an increase in our stroke volume or cardiac output. Exercise will shift our curve up even further to the left because when we exercise, we activate our sympathetic nervous system. As a result, beta receptors are bound in the heart, causing increase in intramyocyte calcium, thus increasing contractility, in a way that we see an increase in stroke volume with increasing end diastolic volume. So as we have increased end diastolic volume, our stroke volume goes up. So really the key concept in the Starling curve is to understand how stroke volume is dependent on end diastolic volume in a normal heart. So we see increase in end diastolic volume, we see an increase in stroke volume. And then also to recognize that in heart failure, these mechanisms are not obeyed, and we see barely an increase in stroke volume with increase in end diastolic volume. And then we see digoxin really helps our failing hearts by increasing intracellular calcium, increase our contractility, and making our systolic ejection fraction better to increase our stroke volume and cardiac output.